unfortunately. Particularly membrane biology, it's a bit old. No, it's textbook stuff I will talk about, but no, there's no issue of recording. Just see whether I can still somehow pull up. Yes, okay. Just that I can also see you guys. In case.
If you just change your slide once, and we are sure that it's also moving forward.
Yeah.
Yeah, it is, and your movies are playing.
Great, wonderful. Okay, fine. Good. Should be good.
I'll mute myself and yeah.
But please feel free to ask and to interrupt. Anything is unclear. Thanks again, Bidisha. Yes, it was a slightly unexpected invitation and short, but always happy to help. And again, I hope it's not totally chaotic what I will present to you, because yesterday evening I quickly put a few slides together. But yeah, we'll see. And again, I'm also not sure with the iGEM. It's a great initiative, whether what I do will fit well into what you try to do, helping some, fighting some rice pathogens, if I understood this correctly. But again, if there's anything that you then would like to know more about, which I might have just mentioned on the side, please feel free to jump in, or at the end of the talk to just ask me a few questions, and I will try to answer them as good as possible. So yeah, as Bidisha mentioned, so my interest is now for a long time already on lipid membranes of cells, of eukaryotic cells. From my PhD onwards, I was very intrigued by how cells actually maintain their stability and integrity when they're exposed to stresses. And then during my postdoc also I went a bit more into trying to understand how actually cells managing to organize the cell membrane and to function, because the cell membrane is basically the interface of the cell with its exterior. So any information, any material that has to cross this boundary has to be processed and somehow has to pass this boundary. And how the cell does this on such small scales is very fascinating. So, and what I will talk here a bit is partly the type of in vitro systems that we use, that I developed during my postdoc and that we continue to use in the lab, but also a sort of another step, how these minimal systems can be used in combination with cells to then understand better cellular processes. So two things, and we'll see depending on the time whether we get to everything. So first, about the motivation, I'm sure as most of you will know, so in your eukaryotic cells, or in general, the cell shape is an important feature. Yeah, it already contains a lot of information, functionality, and in a way, for cells to maintain their shape or to be able to change their shape in an organized way is essential for life. And you can see here two nice examples. One is from the lab of Tim Saunders on the left side, where you see how in the zebrafish, in the tail region, when the zebrafish develops, you have a process where myoblasts, so these are individual precursors of muscle fibers, they start to fuse and then form these striated, very organized myofiber structures that then help the fish later to wag their tail. And one important process here is that the cells, of course, they organize their shape, but also they fuse with each other. So each myotube contains then 2, 3, 4 nuclei. So they're actually the product of multiple cells fusing together. And on the right side, you just see like an example of individual cells, how their shape can actually vary and fluctuate quite quickly. And you can see them crawling around. So these are just like raw macrophages, a cell line that the master student in my lab, we just played a bit around and compressed them, and they sort of freeze, and after some time they can move around again. Just to show to you how sort of flexible the cell surface is. And if you look a bit into the molecular composition, so the two major components that actually control the cell shape are the cell plasma membrane, where you see a depiction, sort of a relative simplification on the right side. So it's a cell plasma membrane that is composed of majorly lipid membranes, but also it contains a lot of membrane proteins and glycans and glycosylated proteins and glycolipids and all that. And underneath, we have a cytoskeletal cortex. So we have a structural element that is important, and that's part of the cell cytoskeleton. I just highlighted the actin filaments here because this is the part of the cytoskeleton that is, I think, one of the major drivers of cell membrane organization, particularly the one that I'm interested in. But just for completeness, also the cell cytoskeleton is made up of three major components, which is actin filaments, then microtubules, which are sort of much bigger structures. So they have 25 nanometer diameter. They're more stiff. They often serve as a kind of long-range transport and organization tool for the cell. And then we have intermediate filaments, which are basically in between the stiffness of microtubules and actin filaments. And that sort of normally are thought to give the cell a general mechanical stability against stretch and stresses, but they don't serve as any type of track for motor proteins, whereas actin and microtubules are tracks for motor proteins. Yeah, but in the whole rest of the talk, when I talk about the cytoskeleton, I actually think and mean the actin cytoskeleton. And that is closely associated with the plasma membrane, and the interplay plays a big role in determining the shape and functionality of the cell membrane. So if you think about that, so this is just try to give a little overview of all the different modes and functionalities that the cell can actually employ using the plasma membrane with the cortex underneath, with the actin cortex. And it's in fact quite interesting that by using, in a way, similar basic elements, we have actin and myosin, for example, in the cortex, and we have lipids in the membrane, but by now having a different sets of adapter proteins at a certain location or certain set of lipids that then activate adapter proteins, the cell can now, in fact, modify the overall organization of this actin cortex to then generate certain structures. Yeah. So we have one of the most prominent ones when cells move around. They have the lamellopodium, for example, where we have local activation of ARP2/3, which is an actin polymerization factor that generates then these characteristic branched actin networks that can push the cell membrane forward. But it is then also accompanied and basically controlled by a local enrichment of certain lipids in the membrane. Yeah? And in this case, it's PIP3 lipids that activate ARP2/3. And then we have a gradient where we have more PIP2 lipids towards the back of the cell, which is a lipid that is more important, for example, activating actin cortex linker, actin membrane linker proteins like S-wins. Or we have can have local activation of formins, and then they can generate parallel actin filaments, which leads to the formation of these pseudopodia. So they have like rod-like protrusions of the cell. And again, you have a very nice interplay then of local activation of actin polymerizing proteins, but also the geometry of the this tube, of the membrane tube, helps also to stabilize this structure of bundled actin filaments. Yeah? And so on and so forth. You can see all sorts of cell shapes and activities are connected to a different way of how it organizes the actin cortex and how the membrane is organized. Yeah. So if we break it down, basically, and that's sort of one of the major motivations for my research is to try and tease out the different elements between the membrane organization, the cytoskeletal organization that lead to this type of interplay and overall can lead to some sort of, you can call it, it's a signaling. Yeah? So there's a sort of mechanosignaling going on, which then leads to either local changes of membrane organization and then local changes of actin organization to allow the cell to do a certain function. And how I do this, I will mention in a little bit. I thought, so one of the approaches to do this, because the cell is of course very complex, is then to use so-called minimal systems where we try to rebuild elements of this complex machinery in a simplified way in a test tube to then try and understand what are the functions of each component by itself, and then to slowly make it more complex to then again reach to something that looks more cell-like. And this is then so-called in vitro reconstitution. Yeah. But before doing that, I thought I give a little bit of background about lipid membranes and how they actually occur and what type of lipids you can find in life, in the world. So the basic question is basically, how do these lipid membranes form and why? And the major part here is already shown in this intro slide, which is the really cool thing about lipids is they're amphiphilic. So that means they have one part, the head group, that is hydrophilic. It prefers aqueous environment. And the tail region, the green one, actually I forgot to ask, can you actually see my mouse, or should I put it as a pointer? I think you can see the mouse, right?
We can see it.
Because if I point by my hands, I guess it doesn't work much. And then you have the hydrophobic part, which is the tail region of lipids. And this mixture of having one element that is sort of that likes to be around water and one element that doesn't like to be water can lead to the self-organization of lipids into a so-called lipid bilayer. Yeah, where if we put this into an aqueous environment, lipids arrange in such a way that the hydrophilic parts are facing the water and the hydrophobic parts are protected from the water. So we can find all sorts of lipids, of course. There's a huge variety of combinations of head groups and tail groups. And first of all, I just thought, how do we call them? And just to give you some examples, I showed you 3 typical lipid structures. Often also these are ones that I use a lot in my experiments. So first of all, as I mentioned, we have this distinction between the hydrophobic tail part and the hydrophilic head group. And so when we name lipids, or when you see a name of a lipid, there's actually 2 parts of it. Yeah? So one, the starting part of the lipid name describes the tail of the lipid, and then comes the chemical description of the head group. Yeah? And so there's sort of the chemical long way of calling them. So for example, here we have diolol S-glycerol 3-phosphocholine lipids. Yeah. So they come from Greek, these nomenclatures. Yeah. And in a way, each chain length has a certain name. Yeah. The dipalmitol says already that both tails are the same. Yeah. So 1 and 2. And then the phosphocholine describes the head group, right? So another way of writing or describing the lipids is by just using these numbers, and where the numbers are in a way describing the tail group. Yeah, so 16-0 means we have a lipid tail that has 16 carbons, and all connections are saturated, so there's no unsaturated part. Yeah. Whereas in this example, yeah, we have an 18-1, which means we have a tail that has 18 carbons, but one of the connections is unsaturated. Yeah. And you might have heard this already. If you think about nutrition, for example, there's always the question, do you have saturated fats or unsaturated fats? And this describes exactly this kind of question. So how many of these carbon bonds are saturated and how many not? And we usually the unsaturated fats, they can react chemically, and they can oxidate, and they can cause troubles. Yeah, so that's why normally the unsaturated fats are not so great for us, and saturated ones are better. And in a way, the fats are actually lipids. Yeah, so if you think about and eating a lot of fatty stuff, to some extent you need to eat them, because some of the lipids the body also cannot really produce, and it is sort of the ground source for the body to make new lipids. Yeah, but of course, if you eat too many and the wrong ones, that also will reflect in what type of lipids will end up in your cells. Yeah? So this is sort of a little hint to metabolism, and it's a huge world, not really my expertise, but just to give you that connection. All right, and so in a way, this is the way how, if you would ever have to then work with lipids, or for example, buy like synthesized lipids, this is how you can then understand, okay, from the name, what is the structure of the lipid, how does it look like? Yeah, and just to give you a kind of a visual idea in a very simplistic way, you can always sort of depict the head group as some kind of circle, quite compact, and then the tails are lines. And again, as a rule of thumb, one can say that the more unsaturated connections you have in your carbon tail, the more spread out that tail will be. So the more kinks you will have in your tails. Yeah, meaning they will need more space. Yeah, they are more brushy. And if you have a fully saturated tail, they will be like straight lines. Yeah, so the lipid doesn't take much space. And we get to that geometric effect a bit later when we think about how our membrane's shaped. Yeah. All right, so I mentioned this already. Of course, now I don't know whether I can actually draw stuff here. I think I can try that. Though, can I make it thick? I don't know. I hope it's so. If we think about having now some lipids, yeah. Let's say I make a lipid mix, and I put it into water. So everything around, yeah, this is all water. What happens now is the tail domains, they want to be away from water, right? And that's why we get something like self-organization, where either, and now I make it a bit easier, I don't want to, sorry. So one sort of way of lipids to organize is in a structure like this, which is called a micelle. Yeah, so where the tails are just pointing towards the center and the head groups are outside. So that's basically the smallest entity you can form. But then if you if there are more lipids around, or if you tweak the way how they can self-organize, yeah, if you make them yourself, you can actually generate bilayers, which means you'll have all heads face outside and the tails face inside. Yeah, and of course, an open bilayer doesn't work, yeah, because then there could be water from the side. Yeah, if such a structure wouldn't really work, because then water could still reach the tails, and it's not good. So energetically not favored. It's not that they decide anything, it's just that it's the energetics that make them rearrange. So you want to have some closed structure. Yeah, so usually you get then some kind of sphere. Yeah, which means then that all the lipid tails are basically protected and just see the other tails, right? So these are the different ways how you can understand why we get actually lipid membranes in an aqueous solution. And to some extent, you could even say, I mean, this is a process that's kind of making these little capsules, yeah? And now you can think of, in this little capsule, whatever is inside in that aqueous solution is basically now isolated from the outside, right? So you can even think in terms of creation of life and how things evolved, that this must have been one of the very first steps in sort of evolution that were important to now allow certain reactions to happen in here, to then amplify their concentration versus the outside, right? So again, and you can find actually lipid membranes in all sorts of organisms, yeah? In the simplest organisms, you will find that. All right. So let's see. back to presentation. Is this any questions also in between?
I have a very naive question, Darius. When you make the supported lipid bilayers, like in the lab, in vitro, then have you checked if you go to the end of the supported lipid bilayer, what is it really? How does it close itself at the edge, or is it really open?
See, I didn't check, but you can think of so this is of course not rigid. So what you'll find, so what I saw, what people think happens, and I don't know whether anyone did EM on that to really see, but you get some kind of so-called frustrated things.
Okay,
like
the
that.
splayed lipids, which they say
So,
wouldn't work, but maybe that's the only solution.
Yeah.
Okay. Fine. Are
It's
there any
quite funny. So okay, we come to that, but because I don't know how many others know what support lipid bilayers where I get to, but when you go, I mean, at least if you use labeled lipids and then you look at the edge, at the rim of your sample, it's quite funny, like get like individual droplets, but I also don't know how much it has to do with there's some other material. No, you have a chamber wall, so there's some plastic as well.
Okay.
I would always, I mean, if you do experiments, I would always try to be relatively central and
Away
not
from
doing it at the very edge.
from the frustrated edges, yeah.
But this is also shown that sometimes you get these splayed things, and so there can be these transition states. And if the system just can't get away from this type of transition state, then well, it's a frustrated. You call it just a frustrated state. And, All right. So just to give you a bit of an idea, okay, lipids, I said you can actually think about all sorts of tail lengths and all sorts of head domains. Now this is just from Wikipedia. At some point I took it, but just to give you an idea, what are the important or the major lipids that we find for mammalian cells? Yeah? And so it's basically phospholipids. Most of them are phospholipids, and then you have these type of major groups like PS lipids, phosphatidylserines, phosphatidylethanolamine, phosphatidylcholines, and phosphatidylinositols. Yeah? And these inositols, for example, they're very important for a lot of signaling processes. Then you have things like sphingomyelin, and you have glycolipids, which are quite important. And then the other part, which is sort of, it's not a real lipid, but it's a lipid membrane. I mean, it's cholesterol, which is sort of not a lipid as such, yeah, but it inserts into lots of lipids and is important again, as you might know. So cholesterol is an important component of all our membranes inside, yeah. And again, that's also the question of what type of cholesterol do you have and eat. All right. So, but if you now think about different organisms, and of course you guys will also work with other organisms, it's actually quite interesting to see how the major lipid compositions change. Yeah? And you can actually connect the variety of lipids and their structure with what type of shapes of do we find and how much do they remodel. Yeah. So in a way, bacteria, you have a couple of lipid types, but it's mainly phosphatidylethanolamine and phosphatidylglycans. They're very robust. Anyway, bacteria often have also like a double membrane structure, and they have also another thing which is called a cell wall, which eukaryotes don't have. And then you have yeast, which is sort of a bit in between. It also has a cell wall, a bit more lipid types, and then you have the higher organisms we already mentioned. You have all these many thousands of lipid types. Yeah. But I just thought, in case you want to read a bit more, you can have a look in this review article. And even within a cell, you will see that, first of all, lipids are not only important for the cell, for the outer cell membrane. Yeah, you have a lot of compartments inside the cell that are also built up of lipids, membrane enclosed compartments. And in addition, it's it's now more and more clear that the composition of these compartments changes. Yeah? So you don't find what we have in the plasma membrane is not the same type of lipids necessarily that you find in the Golgi or the ER. So there's actually gradients of certain lipid types that again have something to do with the functionality of the organelle. Yeah? And again, this is just to give you an idea, like we have the ER, we have the Golgi, we have lysosomes, early endosomes, and they all have a slightly different lipid composition. Okay, this is our slide step 2 now, from the cell now into the lab. What about model systems? And I just thought so. This is just an overview of the typical model membrane systems that people work with, or at least that I know of people work with. Of course, there could be now others. But in a way, so lipid membrane assays were used also for a long time already, because they're not so difficult to make, in fact, yeah, because of the self-organization of lipids, but they're still very, very versatile and much used in science, because they can be used to now again make type of controlled entities where you have a certain composition of membranes of lipids, and so you can test them for all sorts of things. And one of the older ones is like these black lipid membranes. It's basically just ones that you don't image. It's a method often used for incorporating transmembrane proteins, particularly channel proteins. So many people who measure currents of ion gating proteins, like people in neurosciences, they use these type of membrane systems just to have the protein in the right environment. Then we have sort of systems that are flat. So we have the support lipid bilayers, which are in a way flat, planar lipid membrane stuck to a glass slide, where you have it's great for imaging. It's great for turf imaging, for example. It's a nice scaffold to put now other membrane proteins on top or to add cells on top. It's open to optical microscopy and other microscopy methods. Of course, you're stuck on the surface, so you cannot look at membrane deformations in these type of assays. You have to play a bit of tricks if you want to look at transmembrane proteins, because you want to be away from the substrate then, yeah, but there are ways to do that. But it's a very versatile type of easy to realize system. And then we have sort of more freestanding systems, vesicular systems, which are where you can distinguish between giant unilamellar vesicles, large unilamellar vesicles, and small unilamellar vesicles. And the difference here is just the size. So anything above 1 micrometer of diameter is a giant unilamellar vesicle. And they're basically sort of seen as the easiest cell-like system. Yeah, you just make a bag that has roughly the size of a cell. When they're large, you can nicely do imaging on them. Yeah, optical microscopy works in pretty well. They can, proteins and lipids can move freely because they're sort of freestanding. They can undergo membrane deformations, of course. You can incorporate and insert proteins inside that vesicle. So you can, in a way, there's now efforts to really use these containers to recreate basic machineries that we find in cells. Yeah, you can do protein, for example, recombination and expression inside these vesicles and other information circuits and so on. But they're quite fragile, because the lipid membrane alone is quite fragile. Making them can also be a bit harder, depending on what you want to do with them. And then they're slightly easier to make vesicles, which are the large ones and the small ones. Those you can make very, very easily using lipid extrusion. They are often used just to look at, for example, protein binding assays. Yeah, when you just want to see a protein that you made, does it associate to a certain lipid type, so you can easily make this lipid composition and then just see whether the protein binds to these small vesicles and then spin them down. They're very robust, easy to use, but you can't even, you normally can't look into detail what happens on these small vesicles. So the small ones, they're anything between 20 nanometer diameter and 100 nanometer diameter, and everything between 100 nanometer and 1000 nanometers is called large unilamellar vesicles. And then another system that is I didn't work yet much with, but it's also quite interesting, is so-called droplet interface bilayer systems, where in a way you are not working in a aqueous environment. So all the other systems I mentioned are systems where the lipid membrane is formed and moving around only in water. But in this droplet interface bilayers, what you do there is you look at mixtures of oil and water, and at the interface you'll have now the lipid membrane. Again, because of the amphipathic properties, where the head domain will be facing the water side and the tail domain will be facing the oil side. And because you have this interface there, these lipid membranes become very, very stable. Yeah, because they have the support of that oil bath, where there's sort of an unlimited reservoir of lipids available. So if there's any stretching or expansion of area, there's more lipids that can just simply insert, and you have always a continuous lipid membrane. So it can be also very interesting for some assays. All right. Now, I don't know whether you want to have more. So if you want to know a bit more about how to make some of these things, I have another batch of slides, sort of another lecture where I talk about the methods. The other option is I can talk a little bit about the science. So I'm not sure what
yeah, I think the methods we'll do another day. We can go ahead with it.
Right.
But I had a question about the droplet interface, Darius.
Yeah.
Is it not non-biological friendly because you have so much oil around? Or
yeah, so that's one of the drawbacks. If you think about now combining things with these membranes, it is tricky. So one way you can do it and is you can make a, so what you often work with is some sort of emulsion. So you have water droplet inside oil, for example, then the water droplet stays together and it doesn't mix with the oil. So that's what you call an emulsion. Now, if you have lipids around, you can have then a lipid membrane at the interface of this water droplet with the outside. So in a way, if I just want to look at individual cells, for example, what I can do is I can inject in the water already cells, and then these cells will be happy for some time. For some of the gases, they will still exchange with the environment, and the cells will be in the droplet. And then if you, for example, make it flat, you can still generate a sort of lipid membrane that's flat and the cell sits inside. But it's inside this quite limited droplet of a few nanoliters or so. So of course you can't do super long-term imaging, for example. Yeah, you wouldn't be able if you. So there are some people who make networks of these droplets that nearly like form tissues. Yeah? You can make millimeter scale aggregates of droplets that are connected via lipid membranes. But the problem is all around is oil. So you can't just take it and put it into an organism. So some people think, okay, it's a way to make a tissue, but there's this problem that this tissue is surrounded by oil. And if you put it in an organism, the organism will not like it. So these limitations are there, exactly. Yeah. But again, for some questions, it can be quite an interesting approach to generate at least a stable membrane. So that oil layer doesn't have to be very thick sometimes. Yeah. But yeah. All right. Okay. Yeah, so that's just a recall to get back to the biology. So again, now one of the things I'm actually looked at quite a bit is how actin dynamics can lead to local lipid and protein organization. And the in vitro or the simplified model we used is support lipid bilayers to look at that. So this was this work started in NCBS in the lab of Jitu Meyer, and I worked also quite a bit with theorists Kabir Hussain and Madam Rao. And so the question was, or what they were finding already from cell experiments was that it seemed that local ectomycin contractions could lead to the local clustering of certain proteins, of GP, or for example, GPI anchored proteins, or of other actin binding membrane lipid proteins. But it was always too fast and too transient to really capture the structures of actin that would do that in the cell. And so we thought, okay, maybe we can make a simplified reconstituted system that can recreate this type of scenario. So just to make sure that everybody's on the same page. So I was already talking about actin and myosin, but just a little reminder. I hope it's for you guys. So actin is one of these really important cytoskeletal proteins that can form filaments, and it's an ATP hydrolyzing protein. So this globular actin under ATP can form actin filaments. Then you have a polymerization that goes into one direction only. So you have one end that is preferably polymerizing and the other hand that is depolymerizing, and you call the polymerizing end the plus end, and the depolymerizing end the minus end. Yeah, and this whole process also releases ATP. Hydrolyzes ATP. So we have these polar cytoskeletal filaments. They have a certain persistence length, which means they have a certain rigidity. Yeah, it's in the micrometer range, and myosins are motor proteins that work along these actin filaments. And so there's a whole range of different types of myosins. I will not go into detail there, but just to give you an idea, so the major myosin motor that is sort of important for cellular, like global cortex deformations and force generation is the myosin 2. And myosin 2 is a protein. It's about 500 kilodalton. It has two myosin heads. Yeah, it has two head domains and the tail domain. And the interesting thing is these myosin 2 proteins, they can form so-called myosin filaments. Yeah. In contrast to the actin, these filaments don't grow till any length. Yeah, they have a relatively precise or limited size variation. For the non-muscular myosin 2, we have something between around like 12 myosin proteins that form one filament, and these units are then about like 280 nanometers in length. Our muscles, for example, they are also powered by myosin 2, but this is muscular myosin 2, and these myofilaments are much larger structures. So they are normally formed of about 100 proteins, and they can be up to like 600, 700 nanometers in length. And so they contain a lot of motor domains. Yeah. The motor domains, they work again under the hydrolysis of ATP. And here you see the typical kind of cross-bridge cycle of a myosin head. So when they're hydrolyzed ATP, when they have ADP, these myosin heads, they bind to actin quite strongly, and then under the release of phosphate, the myosin head undergoes a conformational change, and that's a so-called power stroke then that pushes along the actin filament. And then the ADP can be replaced by ATP, and ATP myosin would then detach, has low affinity to actin, so it detaches from actin, and then can bind again in a new cycle. Yeah. All right. So the thing is, and this myosin, it sees basically the polarity of actin, so they move or they work towards the plus end of actin filaments. Yeah. All right. So we have a motor protein and we have polar filaments. And so we want to see what can they do if we mix them together. So the in vitro system I used most is the so-called support lipid bilayers. And here's a depiction. So we have a glass slide, and then we form a flat lipid bilayer. We form them by fusing lots of small support SUVs, small unilamellar vesicles. They drop on a clean glass side, and then they will break up and merge with the others to then form one planar bilayer. Yeah, and we can sort of put certain nickel-chelated lipids into the support lipid bilayers, which then allows us to bind any his-tag protein onto this bilayer. Yeah. Now these his-tag proteins, nice thing is because they're binding to lipids, is they can move relatively freely along this plane. Yeah. And that's sort of what would also happen if you have a membrane-binding protein in the cell. And here we used ESRIN actin binding domains that we purified. So this ESRIN is a typical actin membrane linker protein. And then we have purified actin and purified myosin that we can add to the mix. And then we can follow what the system does over time as a function of how much myosin we add, how much actin we add, how much binder we add. Yeah? And we can look at it with turf microscopy, which is a surface sensitive microscopy technique. And so one thing we just looked into is first, what type of actin organizations can we actually generate? And we could see that, and we use short actin filaments with the right amount of myosin. We can generate these small asters, which are these islands of actin that are contracted by myosin action. And this is sort of how it happens. So we have first a quite randomly distributed actin network, and then over time, when the myosins consume ATP and become more contractile, you will see that they will now form these small islands of actin. And now the interesting thing is here, if you look at the actin membrane linker, you'll see that also gets concentrated now in these asters. And that was sort of the first sign of or proof of how actin flows can actually locally increase the density of membrane proteins. And now I see that time is already a bit, it's getting a bit late, so maybe I have to speed up a bit. So this was sort of the first proof of principle for this type of system. We could show that this clustering is in fact really ATP driven and needs the activity of actin. So if you look at the static system, we see a normal Gaussian distribution of clusters, but then if you have an actively contractile system, we can push the system out of equilibrium and find more clusters than we would expect just from random clustering, right? And we could also show that by looking at the correlation between the membrane protein and the actin flows, that indeed where we have more active actin flows, so where the actin moves along fast, we get more of the membrane proteins being dragged along with the actin. Yeah, which was important to show that it is indeed this kind of dynamic effect, the activity that now increases the amount of these membrane proteins. And so based on that, we were then thinking, okay, in a cell, of course, we don't have only one type of actin binder. We have a whole range of proteins that can bind to actin. And how would now a mixture of actin binders behave? And so this was then done by Abra, who's a PhD student, who was sort of my first PhD student in Gitu's lab, who now just finished and defended his thesis a few months ago. And so what he looked into is now he made different actin binding proteins that have different affinities to actin to then see what happens to combinations of two of those actin binders. And the idea here was to see whether there's something called differential sorting or active sorting of membrane proteins. Yeah, because you would expect that or you could assume that the cell might use this kind of activity to now distinguish between different membrane proteins based on their actin affinity. So you made these different actin binders, and to sort of just show one example, here we have one in the middle is a very weak actin binder, and the SN wild type is a very strong actin binder. And now when we have these dynamics, I hope it comes across in the type of that the data connection is good enough to show this to you as well. But if you look at regions like here, you'll see that when we have these actin fluxes now, the weak binder is less abundant. We have these dark spots, and in contrast, you have the bright spots of the strong binder. So you get really this type of local change of mixture. Yeah? And that's just driven by these actin fluxes. And you just see the kind of one example where we cropped out the different time points, and you can nicely see that we get this type of local sorting. Yeah. And so we tried different type of, Abra did try different type of binders. So this is, for example, a strong actin. So two strong actin binders, but one is a bit stronger than the other. Yeah, so you can see the esrin is still a strong actin binder, but utrophin is a little bit stronger. And then you get sort of, they go both together into actin structures, but again, utrophin is much more abundant than esrin. And over time, if you look at the type of sorting ratio, this is the color code here, you would see you get like peaks, like transient increases of one strong binder versus the other. So you could say, this is now a way how the cell could locally, for a short time, say, okay, I really want to have only this one type of protein at this location that might be important for signaling, for example. So this is just a summary of types of mixing patterns you could see. So one is basically where we have complete segregation between the strong binder and the weak binder under actin. Then if you have two strong binders, we can find these type of bullseye patterns. So where we have a strong binder being covering the whole actin-rich zone, and the slightly weaker binder is sort of around the periphery of this, but still enriched compared to the environment. And then if you, as a control, basically, if you have the same type of binder, but we just look at two differently colored versions of the same binder, we get totally mixed systems. And this was then in addition with some theoretical analysis by Amit Das and Kabir, and where they could sort of do Monte Carlo simulations to also explore this type of phase space. The nice thing was that they could also find or predict that we should see this type of different patterns. So, and this whole work is now being written up, and hopefully we'll get this out soon. And I think with respect to time, I would like to keep a little bit of time for questions. I stop here. Stop here and just go very briefly to the current acknowledgements. That's just that you see my lab, get an idea. So I would like to thank, of course, you for your attention. And this is here my current lab in Warwick now, with one in SPE lab member. Maybe if I convince my daughter to also do science later, but we don't know. And so I have my wonderful PhD student, Shantika. She's just finishing up, and unfortunately I didn't have the time now to talk about her work, but then we have Leah and Sabiha, who are currently also PhD students. We have a new PhD student just joining, Badir. And Paulina, at that time, she just came back from Singapore. She did a project in connection with Singapore. And then I have two postdocs. So one is Mobi Selige, who joined last year and works also on the in vitro actomycin network. And then we have a new one who will join, John, who just will be joining in 2 weeks to the lab. And at that, so Deepak was a project student from Bangalore who visited the lab over summer, who did a few experiments with cells, but of course for the whole setup of the startings of the in vitro system, I was at NCBS, and of course I thank Jitu Meyer and Madan, who were really great mentors, and then Amit and Kabir, who were great collaborators on the theory, and Abra and Sankarshan, who are doing the research right now. And thanks to the funding as well, and thanks to you for your attention. Happy for any questions.
Thank you, Darius. It was an amazing talk. Before my questions, let us see if other people have any other questions.
Okay. So, Nivaya.
There is a question.
Yeah,
Shubham,
I
can
read
you
it.
ask your question, or what is happening? I can't read actually very well.
No, I read. It's all right. So it's about the gradient.
Oh, the pip to
Okay,
pip.
why does it help in lamellipodium extension? So it's a good question. So let's put it like this. It was shown or observed that there is this gradient of pip3 and pip2. What we know is that pip3 is basically an activating lipid for the activation of up to 3 and wash and so on. And most likely what happens is that what we know, pip2 and pip3, they're basically locally produced and they're quite short-lived. So what happens is we have these PI3-4 kinases and PI3-4-5 kinases. 1 second, there's someone at the door. Yeah, so what happens, these lipids are actually generated locally by PI kinases, and then they generate this type of gradient. Now, and this then helps the cells to basically activate certain proteins only at that site. What can now be or what is the case in the lamellipodium is most likely that they also have an interplay then of first of all, that the actin network will now limit the diffusion. Sorry. So the actin network will limit and slow down the diffusion of these lipids. Yeah? If you have stronger interacting actin networks with the membrane, these will limit the diffusion of lipids. So in a way, you can now maintain this gradient, a steeper gradient for longer time. And what is also now more likely or shown more and more is that membrane tension plays also a role there. So in fact, it's not only a PIP3-2 kinase lipid gradient, but also that membrane tension is sort of different, and this higher membrane tension at the edge will again help activating certain proteins. How exactly these things are interacting and which comes first basically is not really fully understood. And it's really one of these really fascinating questions. But there were very nice experiments where people start to, by Orion Weiner, for example, with the micropipette, you can suck a little bit the back of the cell to now change the tension of the cell membrane, and you lose the lamellipodium. Yeah. So it's very, very interesting first experiments in this range. But how now membrane tension and PIP3 generation are linked, this is not really well understood yet. I hope this somehow addressed your questions, Sharon.
Any other questions? If not, then I have one. Like when you see the bullseye pattern, what limits the mixing of the red and the green? Are they, in general, like if you look at some other place, are they all? Do they also sort of hate each other, or is it because of the the central one which is easily pushed, that they come and occupy the place first, and therefore the green is cyclic, I mean, the other one is cyclic.
So the nice thing is they don't hate each other from the beginning. So if you look at equilibrium situation, just actin network lying around, normally they're pretty mixed. So you wouldn't even make out any bullseye patterns. So these are snapshots. So these structures, they're not there for all the time then, yeah. They occur and they're maybe maximum when we have maximum flows. And then if the whole structure, for example, is stuck, this sorting will decrease again. And this again has to do with, yes, when you have a strong binder and something is moved along, you have more likelihood that it stays with the actin till the end, basically. And the slightly weaker ones, they lose, yeah, will just unbind and not be dragged that far. And then you have the other effect that to some extent steric type of, if the strong ones already take all the binding spots, then the weaker ones will not be able to follow. And also once the structure is formed, again, the decay of the mixing is actually slower because the dense actin network now limits also the diffusion of the proteins out. So it's really interesting feedback mechanisms. And I know, so it's one of the last things, and I still have to look carefully at that data, but Abra did now some cell experiments where he sort of expressed now differently strong binders in the cell and exposed them to sort of a gradient where they would migrate to then see, and we can see ideas of this global sorting happening.
Nice. Okay. That was a amazing talk. I think just we see very little of these in vitro sort of movies of how molecules can be made to move around or cluster or form asters. Mostly we see either very in vitro in test tubes or cells. So on the membrane in vitro, it was nice to see after a long time. So let us thank Darius again for taking this time out, and hope we'll see him here in person soon.
I'll let you know.
Thank you, Darius. Yeah, thank
Thank
you
you so
everyone.
much. Thanks for your attention. Bye-bye.
Bye. I'll stop the recording.
Yeah. By the way, Bidisha was good.
Bye. Bye, Darius. Yeah.